Alright, today we'll be looking at Watson and Crick DNA structure and how forces interact to stabilize the double-stranded DNA. So first, before we can understand how DNA is stabilized, we need to understand what DNA is. So DNA is a polymer that is made up of many nucleotides, and a nucleotide is made up of a sugar, phosphate group, and nitrogenous bases. So the way we determine if it's a deoxyribose sugar is we look at the 2' position, which would have a single hydrogen, and in ribose we have a 2' position with a hydroxide. Phosphate group is what this looks like. That compared with the sugar, it makes up our backbone, and here's our nitrogenous bases, which are purines are made up of guanine and adenine, and then pyrimidines are made up of cytosine, thymine, and uracil. We won't really look at uracil because that's only an RNA. And this is what a nucleotide looks like when all three of these are put together. So what's the layout of DNA? So this forms whenever we have our complementary base pairing between purines and pyrimidines. This occurs through hydrogen bonding, and these run anti-parallel to one another, which means this side will be 3', this side will be 5', 3', and 5'. So this is 1', 2', 3', 1', 2', 3', 4', 5'. So these are anti-parallel. So as DNA coils up, it forms a helix that can be either right or left-handed. The way we determine which side it is, we look at the sides of the ladder, and these point up to the right. So this is a right-handed DNA. And here we see the major and minor grooves. So what forces actually stabilize the DNA? So we have hydrophobic interactions. That's going to be the main force. That's achieved through base stacking. So as the DNA cools up, the bases are stacked on top of each other. Bases are hydrophobic, which means they push the water out so they can get close to one another. And as they get close, they just really tighten up the DNA. Next, we have hydrogen bonding between the nitrogenous bases through the complementary base pairing. And the more G's and C's you have, the more stable the DNA is going to be because it has more hydrogen bonding. And lastly, we have van der Waals forces, which deals with the how close in proximity molecules are to each other. So what happens to the stability of double-stranded DNA when we place it in a solution with high salt concentration? So as I see here, I've already labeled our backbones with negative charges because it's negative, and the positive ions here is representative of salt concentration. So as we do this, the charges neutralize each other. And as we look over here in our graph, a high salt concentration is going to have a higher melting point than a DNA in a low salt concentration because the DNA is more stable in a salt concentration because the charges neutralize each other. And here are my references.